Well, hello and welcome to our 20th IdeaStream. It's a bit strange to be doing this event virtually, but it's good to see you all via the internet. I know many of you are regular attendees and have come to IdeaStream many, many times. And some of you are first-time attendees. So welcome to all of you. We have many people in Boston. A lot of you are also international or in other parts of the US. Today, we've got a great program, lots of interesting speakers. We've got a fantastic panel to get things going, and talks by our Deshpande Center projects, followed by some breakout sessions where you can actually interact with the speakers. This is our second year of doing this virtually. And hopefully next year we'll be beyond the pandemic and we'll all be meeting in person again. We're going to have a recording of IdeaStream available online in a week or so, in case you missed part of the program or want to watch something again, and we'll put that link up on the website. So today it's a lovely sunny spring day in Boston. And I hope that wherever you are, the weather is also great. This is our health medical sciences day today, and tomorrow we'll be doing our physical sciences projects. You know, to put on an event like this takes money and resources, and I'd like to thank our event sponsors, Gunderson Ditmar, Dow, and Hamamatsu, for their support of IdeaStream. And also in the background, our audiovisual team from Studio 125, led Monica and Keith, who are really working their magic to keep this going very smoothly. And I'd also like to thank the Deshpande Center team of Shirley, Karen, and Eric, who've put a lot of work into this event. So to get started, I'd like to invite Tim Swager, who's the faculty director of the Deshpande Center, to say a few words. Please welcome Tim.
Hello, I'm Tim Sparger. I'm the faculty director of the Sponde Center for Technological Innovation at MIT, and also a member of our chemistry department. I'm welcoming you today to our premier event, IdeaStream, where we highlight our new projects and ongoing projects that have been sponsored by the center. As I mentioned last year during our first virtual IdeaStream, innovation never rests at MIT. In fact, is when we are confronted with challenges, we usually deal with them and innovate around them. Last year, we found that with the pandemic and these new electronic resources that we're no longer bounded by physically having to be in the same room to really engage a broad audience. In fact, last year's IdeaStream had the broadest audience ever from all around the world, with more people than we've ever had in a given IdeaStream. This year, I hope it's equally as large and that we have a lot of repeat customers who come to learn about what's going on in the MIT technological ecosystem. There are many things that we've learned in this last year, and I think have been spawning all sorts of new projects and ideas. We know that the world is more interested in diagnostics at the home, distributed everywhere. We need them. So there are many ongoing and new projects that address, healthcare in the broadest sense. How do we deal with it? I don't want to belabor this too much. I really think that it's most important that we get to hear our innovators. They're brilliant, they're articulate, and they're going to change the world. So with no further ado, welcome and enjoy the time.
Thanks, Tim. And now on with the program. I hope you all have some fruit and drink nearby, because you're not going to want to miss a minute of this exciting show. The speaker bios and the agenda can be found at ideastream.mit.edu. And so let's get going with our panel on funding medical and health startups. Today, our panelists are Christine Bunt and Richard Lim. And Christine has been a founder and CEO of several life science companies, and also an investor in the medical area. Rich has been a venture investor in the biopharma and healthcare space for many decades. So let's invite Christine and Richard to join us. Okay, welcome, Christine. Welcome, Richard.
Thank you so much.
Thank you. Thank you for having us.
Right. So to get things going, in the last year with this pandemic, there suddenly seems to be tremendous interest in healthcare, health-related things, which I'm sure has improved the environment for funding startups dramatically. There's probably much more money, much easier to get funded. So why don't, Christine, why don't you start and let us know, what is the fundraising environment like for medical and health startups today?
It's a pleasure to be here, and thank you for the kind introduction. I think we are in a time which is really unprecedented, and what has started perhaps a little bit dark really facilitated a lot of fundraising, and many new businesses have been started. A lot of funds, and that is publicly known, have actually raised additional capital throughout the pandemic time. And I even went into the National Venture Capital Association database, and we have record data in the year 2020 throughout the pandemic. $166 billion went into startup companies, into 12,500 deals. And we are off to a good start in 2021 as well, with around $69 billion so far into about 4,000, a little less than 4,000 deals. So these are great times for entrepreneurs.
Right. And And Rich, what are you seeing from your perspective in this environment today?
Sure, I'll give you some perspective of what I saw, maybe, and Christine and I may have seen, say a decade ago, where I think getting access and capital for medical and life science startups was, much more challenging. And that was reflected by the numbers of investors who had capital to deploy. I think there were also some technical challenges that were still being, managed through. But I think with the advent of sequencing, some addition of some great new modalities, cell therapies and gene editing, for example. This has really led to a convergence of not only just great technological advancement, but now capital availability. And so today, relative to a decade ago, seeing $100 million Series A rounds is almost getting commonplace, whereas Series A financings of life science deals, I'd say in a 5 to 10, $15 million, maybe a decade ago. So it's dramatic change. I would echo, Christine has described, is the fundraising environment has really improved dramatically.
Great. So there's lots of money around. We've heard that, tons of money available. But the question is, what can founders do to build value before they raise the money? So let's just, Richard, you want to continue? What do you think founders can do?
Sure. Well, I think my advice, and certainly what founders can do, is just stick to fundamentals. Now, think about technical feasibility and how to achieve that. Really thinking about unmet need and a killer app that addresses that. And I think the third and perhaps one of the most important pieces is who are you interacting with? Who's on your team, both as investors as well as on the operations side of things. This cannot be understated how important that is. So those are, I guess, three things. Don't worry about momentum. Don't worry about the market, market conditions and things. Just stick to your fundamentals.
And Christine,
I think what Rich, sorry, I think what Rich is saying is absolutely correct. I don't have much to add other than, try to build your network, meet with people before you even raise the capital. I think that's incredibly important. Yes, we are seeing larger rounds, right? Over $100 million. That's wonderful. But, if you really look under the hood, a lot of those management teams started earlier, working with investors. So, don't be sort of an unknown face by the time you're actually asking for the capital. I think that's something founders, and especially first-time founders, should really invest some time doing that.
So you mentioned sort of the investor side. So let's peel that away and dig a little deeper. And Christine, why don't you start off? What are the top three things that really investors are looking for when they decide to fund something?
Yeah, I think sort of the three things, the magic ingredients, haven't really changed that much. You're looking for a killer team, right? You're looking for a fantastic idea, high unmet medical need in the healthcare field, or a killer application in terms of healthcare IT. And you're looking either for good IP protection or for a very sustainable, groundbreaking competitive advantage. Those are sort of the three magic ingredients. It sounds very, very simple, but it's not. Very, very often you see a pitch where at least one of those are wrong. So it's really important to think about those things early. And if you don't have everything together, again, to your last question, right? Maybe you need to strengthen your team a little bit before you go and raise capital. It's really important to keep focus on that.
Rich, anything to add there?
First, I'll concur everything that Christine has described. Probably the one element I might add is with respect to focusing on differentiation, especially now when it's actually now at a deal frenzy level. The kinds of deals that people are seeing, we can't even breathe. And having a story that really is different sets you apart. Look, many of us we're seeing so many deals; they're almost starting to get blur amongst each other, and so you're particularly attentive when something really catches your attention. It really is around differentiation. We found a new market opportunity. This technology is addressing something that hasn't been addressable for decades. These kinds of things that sort of catch the attention really relate to differentiation. So that's probably one thing I might add.
Great, great. And so, Rich, you mentioned the differentiation. You guys have mentioned there's lots of different money, sort of types of money out there, but what sources of money should the founders be looking for, and why should they go to those sources?
So maybe I'll start on this one. I think be thoughtful about where you're getting the money from, because I think early on, where the decision's going to have lasting consequences, having money that's attached to folks who have had some experience. Now, I want to be cautious not to tell you exactly what to do, but perhaps provide guidance on some of the pitfalls that you might run into, especially around drug discovery and development, which is where I specialize in. Having some early money where folks who have gone through what you're about to go through is incredibly useful. So what I call value-added money. I think where I am cautious is taking money from family and friends who may not have that. There's the personal risk that you are, taking money from folks like that. But I think importantly, you're not getting that sort of vector setting that you can get from folks who have been around a while doing this kind of stuff.
And so, Christine, what about, we hear a lot these days about corporate money that'll come in, maybe non-dilutive, maybe some, rights, a lot of big angel groups. Certainly there's venture capital. What are your thoughts about these different options?
So let me just add to the excellent points Rich made. It is very important that you think a little bit about your capital model when you start something. I mean, this might be perhaps a bit challenging for first-time founders. So again, get a network together with people who have done this before. And I agree, I am an angel investor as well, but you really need to think about how much capital you need to raise to get to revenue point or to an exit, etc. And it can be very hurtful almost, when you sort of dilute the angels out and you're going with private equity or venture capital. You just really have to think that through. To your question regarding corporate money, yes, there are a lot of big deals around, but I often hear entrepreneurs saying, "Oh, we're going to get immediately this corporate deal, etc." You can never count on that. Even if you have the hardest technology, there's so much which can go, I wouldn't say wrong, but which can change at a corporate. I was for 18 years with a large pharma, equally divided between Hoffmann-La Roche and Merck, which are terrific companies. You get a new CEO and you get a new outlook, etc. I also would caution about strategic dilution. You always give something up. This is not just non-dilutive money. It's great to have a good corporate partner, but it absolutely has to make business sense. This is not just about non-dilutive capital. It needs to drive the business or your major product or your pipeline significantly towards the goal of the company. If you're not aligned with that, you actually, a corporate deal can be very dangerous for the business.
Right. So let's get into, we've got a lot of our projects here who'll be looking to spin out companies. Any advice really sort of at the nitty-gritty level of what should the founders be doing? How should they go about raising the money? Some sort of basic steps. Any one of you want to jump in?
I could start,
Pick up.
certainly, and I'll pick up from something that Christine had mentioned already at the outset, and it relates to the network. I hate to say this too, but and you know, who you know does matter, because it links you to people who could help. I mean, this is like any other business. This is not unique to life sciences and, you know, medical. Understanding how the market works, getting to the right vendors, you know, the right syndicate partners, who you know, does matter. So getting out there and developing those relationships is crucial. I will say that the Deshpande Center, and it's not meant to be an advertisement for a Deshpande Center, has been a wonderful nexus point of bringing in outside folks who have helped companies, and they've built, many of the people here built companies with their own hands. This has been a wonderful, sort of nexus point. And I encourage those, soon-to-be founders or current founders to tap the network at Deshpande Center. Everyone plays the Kevin Bacon game. And I think it only takes a couple of steps, if you go through Deshpande, to get to people who can really, truly help you out on a long-term basis.
And Christine, any
Yeah, I
thoughts on
concur
that?
with that. Yeah, I concur with that. Start with the Deshpande Center, but go even further. MIT is a fantastic place for networking. We have so many entrepreneurial professors as well, or students, right? Alumni. So you have a fantastic place to start with, which perhaps other founders won't have. So really make use of that. Another important aspect, which I see at the foundation stage of a company, often there are different founders coming together, and perhaps even beyond MIT, right? So you have Harvard involved, maybe Stanford. Make sure it's very clear where the IP is coming from, if IP is involved at that stage, and sort that out. That's a risk perhaps investors are looking at. There has to be an agreement. So that's value added prior to actually get started with a larger capital raise. Seed funding, that's perhaps okay. The other aspect is, if there are a couple of founders involved, there has to be agreement on the founder shares. Not an easy thing to do, but perhaps a good subject to entertain early on.
Yeah, let me just dig into that a little more, because one of the questions that, gosh, we've been asked many, many times by founding teams is, how should they divide the equity, and does it matter? So we'd love, your experience, your thoughts. What advice would you give them? And Rich, do you want to start with that?
Yeah, I mean, it is case by case, and it is a sensitive subject. So many cases I see folks they just take the denominator and equally cut, the founder shares, for example. So if two founders is 50-50, that, might work for many folks, and that might make sense. But I do think you have to think hard about contributions of IP. Be honest with each other with where how much effort each are going to expend. If you know that you're already spread thin, the expectation of you to take the same share as somebody else who is putting their life work and really working their tail off over the next few years, I think you have to be very thoughtful about that, but put it in writing. I have seen, and it never surprises me now these days, put it in writing, because any post facto changes in agreements or expectations stuff could lead to a really nasty discussion. So put it in writing.
Christine?
I totally agree. This is a very sensitive subject, and it's an art and not a science. I agree with everything Richard has said. Sometimes there it actually helps to have an experienced investor, to have those informal discussions before you even raise the round. They sometimes can help and facilitate a good discussion, and that's needed. Or a couple of very good startup attorneys. They're very well rehearsed in those subjects as well. And I totally agree. Don't fight over this. Agree early. It may need a few rounds of discussion and document it. It's very, very important.
Yeah, and I won't argue, but I will also just add to the people watching is certainly make sure that all the equity vests. It's not just handed out. It takes 4 years before it vests
Very true.
in case people leave the company. We see many situations where it's 2 years in, some people leave, and then they find it's hard to get back all that equity. So looking at, you've seen a lot of startups, seen a lot of founders. Common mistakes that you've seen that have been made by founders. And Christine, do you want to continue on that one? You've been on both sides of it.
Yeah. One sort of, I wouldn't call it necessarily a mistake, but one issue perhaps, which I've seen popping up, is that focus on non-dilutive funding right away. And that sensitivity towards dilution from equity investors, I think that can actually hurt the business. If you do collaborations too early, your economics will be very, very different. I would rather think about creating value, really building your product or your service of your company and bring it to a critical point. And you need some equity financing for that. So I would not be too oversensitive to dilution. And in full disclosure, with my first company I did out of MIT, and I was at the Spande Center project, I was very sensitive about dilution at the beginning with the first round, and I learned my lesson. But it's really important to consider a good capital model to a value inflection point for your business.
Yeah, and Rich, mistakes that you've seen founders make?
I have seen mistakes in really disturbing what might be alignment of interest. I think we're all trying to search for that, and even some of my views about what a VC should do might be counters as to what other VCs might say. But from the founder's perspective, to try to say, for example, in terms of governance, to control everything with regards to the trajectory of the company. This is my company. I want to have last say about things. I encourage temperament around that. And likewise on the VC side, I really don't espouse, documents that give total control to the VC either. This is a very, early stage. We're both in it together. Anything that allows for that alignment of interest, I encourage you, founders to seek that out.
Great. So we're running about 4 or 5 minutes ahead of schedule. We'd normally end the panel now, so I think we've got time for another few minutes of discussion. So we were talking about mistakes. So let me ask both of you. So what are mistakes that you've made and that you wish you'd known about before either starting or investing in companies? So, Christine, any mistakes, or were you perfectly flawless?
Of course, you need to make a few mistakes and learn from it, right? So one of the interesting mistakes, I remember a breakfast at Henrietta's table with Jonathan Fleming, and he wouldn't mind me giving this as an example, where I delivered a pitch for a capital round. I forgot even which company it was, and he said, stop right here. I said, yes, is something wrong? I want to really learn about you first as a person, rather than, your pitch and your business model. So it comes back to what I said earlier. We as investors, I'm switching my head now, I'm going from entrepreneur to investor. We want to know about the people, what you can do, because we need to project into the future, do reserves of capital, etc. Is this the team? Is that the person who can take a concept and build a product, in a couple of years down the road? So I really underestimated, and perhaps coming from pharma, which is still a fairly conservative environment, how important it is to get to know people and let investors know about your personality and how you approach problems, etc. So that was one of the mistakes I made early on.
And Rich, I know you've never made any mistakes.
I made tons of them, and that's the only reason I think why we are where we are is because you hopefully have learned from some of those mistakes. I think probably the one principle that I continue to monitor for myself is if you see something that's wrong, whether it be a factual item and or your intuition is suggesting something is wrong, address it. Try to do it obviously in a productive, constructive manner. And I really emphasize that. I just see so many egos, get involved, but shoveling it out to the future, you're going to pay for it later. So if you see a problem, be thoughtful enough, and don't worry about deference or hurting feelings and stuff. Again, if you do it thoughtfully, the other side should find it, highly enlightening that you brought it up. Because I think both parties would, founders and investors would really suffer from that. So again, be honest with things that strike you as being problematic right up front.
Right. Well, it's been a great panel. I know we could go on for quite a long time having this conversation. And I want to thank both of you for spending the time with us today. And I'm sure people have specific questions, they will reach out to you. But Christine, Rich, thank you very, very much. And thank you also for all the time you spend, as catalysts, mentors at the Deshpande Center, because you've both been involved with us for quite a long time. So thanks very much.
Thank you very much. It was a
Thank
pleasure.
you. It's been an immense pleasure. It's an immense pleasure.
Okay. So now we're on to the part of the program where we're going to see some of the interesting technologies that get developed at MIT. As everybody knows, MIT does fantastic research. And our focus at the Deshpande Center is really on commercializing that research, getting it out into the world where it can have an impact. And what we do is we provide grants and mentoring to faculty and their teams to really try to move their technology to a point where it can spin out, where people like Richard and Christine would actually fund it. And all the presenters that you'll see in the next 2 days are our grantee teams. And what you're going to get is really a window into the future from some very, very bright young people. And what we'll do now is we'll have 5 presentations, and then we'll have a break where you can meet with the presenters for questions and answers. We have a great speaker to kick off the day. Linda Griffiths is a professor of biological engineering at MIT, and she's going to tell us about her technology to improve the process for storing human eggs, which will help women who wish to postpone pregnancy. Please welcome Linda.
Thank you so much, Leon, for that wonderful introduction. I'm here to share with you today our vision for revolutionizing female reproductive health care by bringing together teams of clinicians, mechanical engineers, electrical engineers, biologists, together even with patients, so that we can bring mesofluidic technologies developed at MIT directly into the care of patients. We start with an observation that the maternal instinct is a force of nature. You cannot fight it. The most calm, collected, organized, professional woman can become a bundle of irrational emotion when she can't get pregnant or she has diseases that affect her fertility. So at MIT, we decided to focus on 2 of the most challenging problems, those in fertility preservation or egg freezing, and those in bringing personalized therapy to certain gynecology conditions that afflict about 10% of women. We started on this almost 15 years ago. So this technology developed over a long time. We started when a local gynecology surgeon, Keith Isaacson, approached me saying there were no options for the 23-year-old he had just operated on for her 10th surgery. What can he do for the patients who can't get pregnant or get relief from the pain and suffering of the gynecology diseases? So we got a foundation grant, and since then started working together. Since then, we published seminal papers on endometriosis molecular classification and won a number of awards. Let me share with you the technologies that we're now ready to bring into the clinic that have been percolating for all these years. One is in the problem of egg freezing. The issues are a graduate student today who's worried about will she be able to get pregnant and wants to freeze her eggs because she goes and works at Facebook, which has amazing fertility benefits. Will it even be reliable? Will she even have access if she's working remote to a clinic? And furthermore, if she has a disease, will she get holistic care? So what we decided is the huge challenge is that so much of the process is manual. A reproductive endocrinologist manually takes the eggs and puts them into a cryopreservation fluid and then freezes them. So this is highly susceptible to operator error. And it's also very expensive to do this. So we want to reduce the cost and make it more accessible. So we're bringing technologies from Scott Manalis's lab, where he has made tremendous advances in technologies to manipulate individual cells, measure them, characterize them. And we're now bringing this kind of technology into manipulating the egg so we can automate the egg freezing process, thereby making fertility preservation accessible to all of the light blue areas on this map, and also reducing the cost and time required to go through the process. But if a woman is healthy and she can freeze her eggs and get pregnant later, that doesn't help her if she has endometriosis or adenomyosis, two very common gynecology diseases characterized by having the uterine lining growing outside the lining of the uterus, either in the wall of the uterus or in the abdominal cavity, causing terrible pain, infertility, anemia. So we've been pulling together approaches where we actually go in and characterize how patients are different. What is the molecular basis for differences in patients? What are some of the other differences in patients that might let us bring a more personalized care to these patients? And what we were told over and over again by pharma companies is that we needed approaches that would let us represent the patient in our lab to test new therapies. So after characterizing some molecular differences between groups of patients, we started to build new tissue engineering technologies to actually grow individual patients that we know something about in the lab. This is showing some proprietary tissue engineering approaches that we developed where we can grow the not only the patient organoids, but a more complex model of several cell types representing the lesion in the lab. It's one thing to grow these in standard kind of incubator format in a 3D culture as we show here using some biomaterials we developed, but it's another thing to have them be more representative of a lesion by having blood vessels and nerves and so on. So we've teamed up with Professor Roger Kam, who has developed some beautiful technologies to make microvessel networks grow in microfluidic devices. And what we're doing now is making these a much more robust approach to go to a larger scale so we can grow an actual lesion, and so we can simulate a patient's menstrual cycle in the lab. This is building on technologies out of a big DARPA program we led for 7 years. And what I'm showing you here in this movie, these are onboard pumps. These are tiny microfluidic pumps that can serve like a little beating heart to circulate fluid through that kind of vascular network. Not only that, it can circulate the fluid, but it can then allow cycles of hormones to mimic the menstrual cycle. So what you saw were the pumps. Now you see us building the patient. This is bringing that tissue engineering approach directly onto this chip so that we can keep it going for weeks at a time and study the outcome. So we look forward to talking with you at the end of the session today about how we can make this outcome shown here. This is actually my niece who has endometriosis, and she had a baby, and we hope we can make this happen for all who want it. I look forward to seeing you.
Thank you, Linda. Great talk, interesting technology. Our next speaker is going to be Anna Jagilska, who is a researcher in material science at MIT. And Anna will tell us about her technology that will allow better screening of drugs to treat myelination-based neurological diseases like multiple sclerosis. Please welcome Anna.
Hello, everyone. Thank you very much for this introduction. I'm very happy to talk to you today about artificial axons, a novel biofidelic drug screening platform for neurological diseases. Neurodegeneration is a growing problem. There is nearly 1 billion people affected by some type of neurological disorders. These disorders often lead to neurodegeneration, where neurons die, leading to disabilities. Unfortunately, today we have no cures for many of these neurodegenerative diseases, including Alzheimer's, Parkinson's, and multiple sclerosis. Although these diseases are diverse, they have one feature in common. They often lead to damage of myelin. And myelin is this very important membrane that is wrapped by cells called oligodendrocytes around axons of neurons. Myelin works like an insulator, allowing neuron to conduct signals. And when myelin is damaged, neurons can't conduct anymore and die. Therefore, a key strategy in preventing neurodegeneration is stimulating myelin repair in the process called remyelination. Unfortunately, today we have no drugs available for remyelination. Part of the reason is that we also don't have adequate tools to study myelination and screen for remyelinating drugs. In order to do that, we need axon-like structure, a thin fiber with very low stiffness. So what is available today to study myelination? We have assays that are based on tissues and co-culture, which provide similar to in vivo environment, but they are too complex for drug screening. On the other end of the spectrum, we have tissue culture plates, which are very simple and reproducible, but they are flat and extremely stiff, therefore biologically irrelevant. Therefore, there is a need for three-dimensional solutions that will be biofidelic and at the same time reproducible and scalable. To fill that gap, we have engineered artificial axons. In this image, each fiber is made of a polymer and represents biological axon. It provides axon-like geometry and stiffness, and we can make those constructs in a very reproducible and scalable process. To create our solution, we have developed an advanced 3D printing technology. We provide axon-like geometry with micrometer scale diameters and stiffness below 1 kilopascal, matching that of biological axons. Our platform is reproducible and drug target agnostic. It can be used for many classes of drugs. It is also tunable. We can change the geometry and properties of the material, which allows us to model different diseases. We have demonstrated that artificial axons are biocompatible with neural cells, and you can observe directly myelin wrapping and quantify that process. As an example, you can see here green myelin membrane wrapped around artificial axon. The platform is scalable and compatible with standard laboratory practices, including live imaging of myelin wrapping. As the first commercial application, we focused on myelination assay because it is critical for development of therapies for neurodegeneration. There's also significant market in that sector, and there is no cures for remyelination. Artificial axons provide a better myelination assay. It is the only assay with quantifiable direct readout of myelin wrapping. You can see here in the image green wraps of myelin around artificial axons. You can quantify this process as a function of different drugs and doses, and our platform is capable of measuring those responses. Artificial axons are also cheaper and faster than current assays, and they are reproducible and suitable for high throughput applications. With recent advances in stem cell research, human-derived neural cells are becoming a standard in the development of neurological therapies. With access to individual patient cells, we are learning that neural system of each patient is unique. Therefore, personalized approaches to therapy development are needed. Artificial axons are ready for this next challenge. They are the only high-throughput myelination platform that is compatible with human cells. Using artificial axons, we are able to capture differences between patients in myelination, which opens opportunities for applications such as patient-optimized therapies or patient stratification in clinical trials. Key highlights of our technology include customization. We can generate any geometry and we can tune material properties, which allows us to model different diseases. The scalability, we can generate individual samples or the entire plate of samples. The high throughput, we can make that plate in minutes. And high resolution, we can generate micrometer scale features, providing high level of biofidelity in our models. With this versatile and flexible technology, we plan to expand interest of our future company beyond drug screening to disease models, drug disease mechanisms, biomarker discovery, and brain organoid research. Our platform provides a more predictive in vitro assay, which if used early in drug development process, it can reduce cost and time of drug development. We have validated our platform via strategic collaborations with multiple academic partners and also industrial collaborators. We plan to launch a company in the fall of 2021, and our business model relies on building drug discovery alliances with pharmaceutical partners and co-developing of therapeutics. We have been working on artificial axons for many years with a wonderful team of researchers with expertise in biomaterials and 3D printing and fantastic business advisors. I want to acknowledge our collaborators and funding and finish by inviting you to the Artificial Axon booth. We are launching in the fall of 2021 and are currently searching for CEO advisors, seed investment, partnerships with industry, and also passionate potential new team members. So I hope to see you at Artificial Axon booth. Thank you very much.
Well, thank you very much, Anna. And so any of you who can help her as they start the company, want to get engaged, as she mentioned, be you investors, collaborators, team members, please do reach out to her and go see her also at the breakout booth. Our next speaker is Chelsea Catania. Chelsea's a postdoc in mechanical engineering, and she's going to talk about an encapsulation method to enhance the microorganism viability for medicine and food supplements. Please welcome Chelsea.
Thanks for that introduction, Leon. My name is Chelsea Catania, and I'm working with Professor Ariel First in the Department of Chemical Engineering. And I'm excited to share with you our project on keeping microbes alive. You might be wondering, why would we want to keep microbes alive? We were raised to think that all germs are bad. They're everywhere and they should be destroyed. But that's not the case. We're recognizing now that many bacteria are beneficial to our overall health and well-being. And many of them are actually pretty difficult to grow and keep alive, which is why we need to develop methods to keep the beneficial microbes, or our probiotics, alive and well. Consumer demand for probiotics is huge. The market is large and rapidly increasing. And while there are many products available, there aren't many bacteria species to choose from. Most probiotics on the market come from just 4 groups of bacteria, which also means their health benefits are limited. There's a huge opportunity right now in identifying other microbes that have new and even therapeutic benefits. This is obvious when we look at all the microbiome therapeutic companies that exist today and their valuations, in addition to all of the new startups that keep popping up across the world. Most, if not all of these companies are focused on discovery. And although the future is promising, no new products are on the market yet. That's because these next generation probiotics are currently impossible to manufacture. Most of these target microbes are sensitive to oxygen, which means they need special handling, else they die in the process. Not to mention all of the mechanical, thermal, and chemical stresses that occur during manufacturing and in storage that affect the viabilities of all kinds of probiotics. We're developing a protective coating that can self-assemble on the surface of each cell while in solution. Unlike existing methods, this can be done on any type of cell and provides single-cell protection from manufacturing stresses. It's real simple. We just add 2 components to solution for the coating to form, which also makes this method easy to scale up and deploy in existing manufacturing lines. You can think of our tech like a tiny suit of armor for bacteria that helps them make the stressful journey from the manufacturing floor to your gut and ready to fight for your health. Existing industrial processes generally follow this scheme. Bacteria are grown to a certain density and then concentrated. Growth media is replaced with a formulation specific for freeze drying, and then the dried product is put into pills. Our method can easily be integrated with the formulation step, enabling us to bring currently intractable microbes to market. One example of a currently intractable species that shows great promise for both humans and for animals is Faecalibacterium prausnitzii, or F-Prau for short. High amounts of F-Prau in the gut directly relates to overall health. However, these bugs are extremely oxygen sensitive and die within minutes when they're exposed to air. Extending the time this microbe can be handled would be monumental for its manufacturability. And this is just one example. Our method can protect all kinds of microbes from different stresses, not just oxygen. So far, with the help of the Deshpande Center, we've developed the coating process and demonstrated we can coat a handful of different bacteria species using no special equipment. We've monitored the viability of cells after the coating process and tried a few different chemistries to enable better cell growth. We also demonstrated that our material coating remains intact after freeze drying. Now that we've honed in on our technique, we're ready to test it out on F-Prau. We'll be looking at how well our method protects this bug from air while it's in solution, after freeze drying, and after some time in storage. In summary, we're very excited about the future of microbiome therapeutics, and we want to enable this industry. Our goal is to produce a final product containing high levels of viable microbes. We focused on air tolerance because that's the biggest problem right now. But the material properties of our coating would enable our armored bacteria to withstand all sorts of stress. In the end, this method could be applied to a wide variety of microbial products across various industries. Last summer, during both the NSF I-Corps program and other entrepreneurship programs, we conducted over 160 customer discovery interviews across many different industries, which led us to this hair on fire problem in microbiome therapeutics. But this technology is applicable to nutraceuticals, agriculture, and even cosmetics that employ microbes. With the Deshpande Center's support, we've been able to develop our method. Once our provisional patent is filed, we'll be able to search for development partners who have other microbes they want to bring to market, and we'll be seeking non-dilutive funding sources to continue our work. We're currently looking for microbiologists or industry members to collaborate with or team up with. If there's a microbe you want to protect, we'd love to talk to you further in the breakout room. And with that, I'd like to acknowledge our team, Professor Ariel and Dr. Gong Fan, and the Deshpande Center members for their advice and support. Thank you.
Thank you, Chelsea. Great talk. Our next speaker is going to be Dion Lee. Dion is a postdoc in the Research Lab for Electronics, and he's going to tell us about a point-of-care system for monitoring sepsis in patients. Please welcome Dion.
Morning, everyone. My name is Do-Hyun Lee, a research laboratory of electronics at MIT. And today I'll introduce the Deshpande support project, which covers the developing a point-of-care integrated microfluidic platform for sepsis monitoring. The project is advised under Dr. Joe Volkman. Sepsis is a clinical syndrome characterized by systemic inflammation that arises from a serious bloodstream infection. Sepsis has a high healthcare burden for nearly 24 billion in annual cost, and is the most expensive condition to treat in U.S. healthcare system. Also, it is the 11th highest cause of the death in the U.S. and has 18 million sepsis diagnosis cases, with the incidence of this condition increasing by 10% annually. Clinical studies show that the mortality is significantly reduced if sepsis patients are identified and treated at early stages of the disease process. The key element in treating severe sepsis include early antibiotics, early recognition and intervention, source control, and hemodynamic support. Also, sepsis is a fast-moving disease as a clinical emergency where patient state can change substantially in a matter of hours. Therefore, clinical monitoring with frequent assessment is the most important among severe sepsis patients to reduce the mortality. So after diagnosis, sepsis patients are commonly monitored by clinical criteria, vital signs, and peripheral blood glucose counts. Based on the monitoring results, then we can expect to the potentially better titrate medication therapy, which leads to reduced lengthy treatment and healthcare costs. Okay, we are assuming that you are already in the intensive care unit, which is ICU. You will do the CBC, which is complete blood cell count, and this provides a census of the immune cells from your blood. And this is a super important assay, but it requires lots of blood draw over 3 milliliters, several hours for sample preparation and isolation. And it doesn't tell you what the cells are doing, and it doesn't tell you the function of the immune cell. Also, the whole process is manual and labor intensive, as well as the rapid sampling for the CBC can lead to blood loss. Among the type of white blood cells, here we are looking at the neutrophils that help heal damaged tissues and resolve the infections. So neutrophil blood levels increase naturally in response to infections, injuries, and other types of stress. Also, bacteria allows the neutrophils to become activated during sepsis. That's why we are focusing the level of activated neutrophils versus non-activated one. So we have a straightforward pipeline that to have patients take a very small amount of blood and take the white blood cells out at the first stage, and profile the white cells using the small microfluidic device with electronics that will be described at the next slide. So we have a microfluidic device in here. The cells are coming in the one side, and they enter this device and encounter these electrodes here, and the electrodes energize the small voltage that creates the little electrical force in the cells, which push them across the channel. So the activated and non-activated neutrophils can be deflected and differentiated to each lateral position in the microfluidic channel, and the results end up the distribution of the cells like this histogram. So again, the platform is only 50 microliters of whole blood and 25 minutes assay time. And we have done the pilot study at the Brigham and Women's Hospital. And as a result, the point is that if you look at the number of different samples across healthy and sepsis patients, you get a slight separation between two states. When you look at the correlation between the total amount of neutrophils by CBC and the clinical score called SOFA, 0 is healthier and 20 is very sick, you can see a quite spread, but when we took this data in terms of the microfluidics has a best correlation to the clinical score. So we are pretty excited about that. It has potential value as clinical indicator of the immune system. As support from MIT Desk Funding Center, we would like to provide a fully automated sepsis monitoring solution which can be reliably run by ICUs, clinics, and hospital labs to improve sepsis care. We are now working on reduction time to 15 minutes assay time, lower the sample volume below 50 microliters, which can translate from blood draw to finger stick, increase the sensitivity and specificity, lower the test costs, and developing the user-friendly system to enable bedside assessment. While we focus on here on sepsis as a well-defined and clinically important use case, our system has applicability beyond sepsis, such as pediatric immune disease, coronavirus, and autoimmune diseases, and so on. In the current coronavirus crisis, real-time immune monitoring using IDS could also be useful for adjusting treatment in coronavirus-infected patients. Also, treatment of chronic autoimmune disorders such as rheumatoid arthritis could be placed at the patient home for routine monitoring of disease. Thank you so much today, and I hope we have a great discussion during the network session, and please reach out me and Dr. Joel Boltman if you have any questions and comments on this project. And I'd like to thank you, our collaborators at Brigham and MIT and the MIT Dash Fund Center. Thank you.
Thank you, Dohan. Our next speaker is Michelle Dion. Michelle is a PhD student in health sciences and technology at MIT, and she's going to tell us about a treatment for glioblastoma using a localized drug delivery technology. Please welcome Michelle.
Thank you for that introduction, Leon, and thank you all for joining me today. My name is Michelle Dion, and I'm a third-year PhD candidate in Professor Natalie Artzi's lab in the Institute for Medical Engineering and Science at MIT. Today, I will discuss our team's research into treating glioblastoma by delivering therapies directly to the post-surgical resection cavity using our adhesive hydrogel technology. So, despite significant advances in cancer therapy, brain tumors continue to be one of the deadliest and costliest of human diseases. Today, we'll focus on glioblastoma, which is the most common and most lethal type of primary malignant brain tumor in adults. Current glioblastoma therapy consists of surgical resection followed by concurrent systemic temozolomide chemotherapy and radiation. Surgery removes the bulk tumor and provides clinical relief. However, it is difficult to remove all cells using surgery alone, as tumor cells diffusively infiltrate healthy tissue. Thus, radiotherapy and chemotherapy are used to eliminate these remaining cells following surgery. However, almost always tumor cells persist and drive cancer recurrence. This results in a dismal prognosis for patients as indicated by a median overall survival of 15 months and one of the lowest survival rates of all cancer types, less than 10% over 5 years, as shown in the curve on the right. One of the major reasons why current therapy fails is because it is unable to effectively be delivered to the brain tumor. A key challenge preventing systemic therapy from reaching the brain is the presence of the blood-brain barrier. The blood-brain barrier segregates peripheral blood from the delicate brain parenchyma, providing protection from circulating pathogens and toxins. While this barrier function is important to brain health, it also renders the blood-brain barrier impermeable to an estimated 99% of therapies, severely limiting the treatment options available for this challenging disease. Furthermore, many drugs that cross the BBB exhibit poor permeability, preventing the accumulation of drug in the brain at sustained therapeutic levels. Poor accumulation drives the need for increasing the amount of therapy used, which in turn increases the accumulation of drug in off-target tissues like the liver and spleen. This can result in the development of adverse side effects due to off-target toxicity. Even for therapies that successfully reach the brain without causing systemic toxicity, additional challenges remain. These include delivery challenges like drug transport and retention within the brain tissue and specificity of drug uptake by tumor or target cells, and biological challenges like intratumoral heterogeneity. Our approach to these delivery challenges is to use an adhesive hydrogel to mediate controlled delivery of therapy to the post-tumor resection cavity or inoperable brain tumors. Our hydrogel can serve as a local depot for a variety of therapy types and can provide tunable sustained therapy release. By delivering therapy locally, we are able to circumvent the blood-brain barrier and confine exposure of the therapy to the tumor site, limiting off-target toxicities and reducing the dose required to achieve efficacy, addressing many of the challenges I spoke about on the previous slide. Our hydrogel consists of two components, oxidized dextrin and polyamidoamine dendrimers, shown here. Upon injection, the aldehyde groups on the dextrin polymer can react with either the amines on the surface of the tissue, shown in pink, or on the surface of the dendrimer. This is a competitive reaction by design, where the tissue amines have faster reacting kinetics with the aldehyde groups than the dendrimer amines do, allowing tissue amines to be consumed first. This imparts tissue adhesion, which allows the material to mold to the tumor cavity to achieve high drug-tissue interfacial interactions. The remaining aldehydes are then consumed internally by the dendrimer amines, imparting hydrogel cohesion and controlling drug delivery release. By modifying the formulation of the hydrogel, we can thus tune its mechanical and drug delivery properties to meet clinical needs. In this slide, I've summarized the technical features of our hydrogel that make it advantageous for local delivery to the brain. On the top, you can see examples of how hydrogel formulation can dictate therapy release kinetics and its responsiveness to the disease microenvironment. We've also demonstrated that the hydrogel can be sprayed onto tumor tissue to form a cohesive gel that is scratch-resistant and immersion-resistant, making it ideal for administration to the post-surgical cavity. In preliminary intracranial in vivo studies, we've also seen that the hydrogel is well tolerated and biodegradable. We have shown the versatility of this hydrogel technology to mediate controlled delivery of small molecules, nucleic acids, and biologics to peripheral tumors over the course of hours to weeks in our recent publications. To generate proof of concept data in intracranial tumors, we are conducting ongoing efficacy studies in orthotopic tumor-bearing mice. In one of our studies, we are examining the effects of local doxorubicin chemotherapy in comparison to systemic temozolomide. Systemic temozolomide chemotherapy represents the current clinical therapy and is dosed daily over the course of 5 days. Doxorubicin was chosen as it is an effective and well-established chemotherapy that currently cannot be given to glioblastoma patients due to its toxicity. Here it is formulated with our hydrogel and given via intracranial injection into the tumor bed. The mice are monitored for tumor burden via bioluminescence IVUS imaging and for overall survival. The plot shown here depicts the bioluminescence of the tumor, a measure of tumor burden in this model over time. You can see that following hydrogel injection on day 10, tumor bioluminescence decreases for groups treated with doxorubicin-containing hydrogels in comparison to untreated control groups and groups treated with systemic temozolomide alone. Here I've added representative images obtained using IVUS for mice in the untreated control group on top and in the doxorubicin-loaded hydrogel treated group on the bottom. Control mice show intense signal on day 20 and soon succumb to tumor burden, whereas the majority of mice treated with doxorubicin hydrogels show resolved tumor bioluminescence signals, suggesting decrease in tumor burden as a result of therapy. This reduction in tumor burden manifests in increased survival in mice receiving the doxorubicin hydrogel therapy. Whereas all mice that have received control or oral TMZ therapy have died, the majority of mice that received hydrogel-mediated dox therapy remain alive and will continue to be monitored to see if tumor recurs. While the study is still ongoing, preliminary Mantel-Cox analysis shows that the survival of the doxorubicin treated group or the combination therapy group is significant in comparison to control or oral TMZ groups. In summary, the results I have shown today demonstrate the promise of our local hydrogel delivery technology to circumvent existing delivery barriers and mediate effective glioblastoma therapy. We are currently investigating different drug candidates, including targeted therapies and immunotherapies, and different hydrogel formulations in vivo in order to select a lead drug candidate formulation. We welcome the opportunity to partner with you to expand your therapy's impact in glioblastoma and beyond. With that, I'd like to thank my entire team and thank all of you for listening to how we are expanding the therapeutic arsenal available to brain cancer patients by improving intracranial drug delivery using our adhesive, tissue-responsive hydrogel technology.
Well, thank you, Michelle. And thank all of you speakers in our first session. You've done a great job. And you certainly kept within the 6-minute time window that we asked you to do. So we're actually right on time, even a few minutes early here. So now we're going to go to the interactive part of IdeaStream, where you, the attendees, are going to be able to interact with these speakers in breakout sessions. The way we're going to do this is we're using Hopin's Expo booth feature. And if you look on the left-hand side of your screen, you'll see Expo. If you click on Expo, you'll find the booths. And what you do is then you click on any booth to enter it. And you can go from one booth to another. So you can leave a booth, go to another booth. And to turn on your audio and video, when you're in a booth, you click Share Audio and Video, and then you can interact with the speakers. You can also use the chat function once you're in the booth to talk to them via chat. Now, a lot of people can be in a booth at the same time, so that's more or less unlimited. However, only 10 can appear on camera at the same time. It's as if you were at a poster session, you'd be waiting in line to talk to the person at the poster. So if all the on-camera slots are taken, then just wait a few minutes for that conversation to wrap up and then request to appear on camera again by clicking the Share Audio and Video. And certainly, again, encourage you to go talk to the various speakers, ask questions. There'll be other members of their teams in the booth. They can share information with you. Certainly go back from booth to booth. What we'll do, it's now, let's see, in Boston, it's about, according to my clock, it's about 12:06, 12:07. So we will meet back here at around 12:30 Boston time. So that's another 24 minutes from now. And the way you do that is this is the stage. So you'll just click on the stage in about roughly 20 to 24 minutes time. And we will continue with the rest of IdeaStream. So enjoy your interactions with these really interesting projects and interesting speakers. Thank you. Well, welcome back, everybody. I hope you enjoyed the break, had some good conversations with our presenters, and maybe got a chance to get something to quickly eat and drink, and are ready for the afternoon session here. Well, while we were doing the show, I got this email here from it says from Mirna in Boston, and Mirna said, this has been a really long pandemic, and I'm tired of cooking the same things every day. Don't you have some suggestions for me? But I don't really want to spend a lot of time. So, Mirna, I have for you a fantastic traditional South African recipe, which is very fast, it's very easy, it's delicious. It's South African chicken thighs with chutney and mayonnaise. You can also look up these recipes on the internet. Just look up South African chicken thighs, chutney, and mayonnaise. So this recipe, it's just 5 ingredients. First, you need chicken thighs. So you have some chicken thighs, chutney, mayonnaise, onion, and garlic. So here's what you do. Very simply, you start out, heat your oven to 350 Fahrenheit. For those of you overseas, that would be 180 degrees Celsius. And put your thick chicken thighs in a baking dish on the counter. Take the onion, chop up some onions, put them in a frying pan and just cook them for about 5 minutes in some oil. Then chop up the garlic, throw the garlic in for a minute. You don't want to go much longer than a minute. You don't want to really burn it. Then you add the chutney and the mayonnaise into the pan, a little bit of salt and pepper, mix it all around. And then you spoon that mixture onto the chicken, smush it around on the chicken, stick it in the oven, and just bake it for about an hour until the internal temperature reaches about 100, or at least I'd say 180 degrees Fahrenheit, or in Celsius that would be about 80 degrees Celsius. And trust me, Mirna, it will be delicious. You'll love it. Your family will love it. They'll think you're an ace cook, and your kids will definitely love it. So, hope that answers your question for today. And now let's go back to some of the Deshpande Center stuff. And for those of you who don't know a lot about the Deshpande Center, here's a brief history. We've been around about 19 years, and during that period, we've given out over $20 million in grants to 170 projects. From those, we've seen 43 spin-outs, and those spin-outs have raised over $1 billion in capital. And one of the keys to our program are really all of our catalysts. These are the mentors, a lot of whom are in the audience today, who volunteer their time to help and mentor our projects. And I want to thank all of you for your tremendous time, commitment, and dedication to the center and to the teams. The Deshpande Center is a self-supporting organization at MIT. What does that mean? It means we don't get funding from the central body at MIT. We essentially raise our own money, and we sustain the center through philanthropic support, foundation support, and also corporate support. And we want to thank all of the philanthropic supporters for their tremendous generosity and trust in us. We know there are other very worthwhile organizations you could be supporting. I also want to thank the Alana Foundation, who have funded our Technology to Improve Ability program, and also corporate members of the Deshpande Center, IMA, Henkel, Lockheed Martin, and Sanofi for their really long-term financial support of the center. And any corporations that are interested in engaging with us, please reach out and contact us, and we can tell you how you can become more engaged with the Deshpande Center. So now we're going to move on to the second set of speakers. We have 6 speakers coming up now. Again, some very, very cool, interesting stuff that you're going to hear. And our first set of speakers are from our Technology to Improvability program, and they are Christy Johnson and Jaya Narain. They're both PhD students in the Media Lab, and they're going to talk about a communications technology to help caregivers understand people who have trouble speaking. So please welcome Christy and Jaya.
Thanks, Leon, for that kind introduction. My name is Jaya Narain, and I'm here with Christy Johnson. We are PhD students at the MIT Media Lab. Today, we're going to share our work on Kamala, which is a play on the words communication for all. This technology is working towards improving understanding of nonverbal vocalizations from non-speaking individuals. Over 1 million people in the US are non-speaking or minimally speaking. In particular, our work focuses on individuals who have 0 to a few dozen spoken words or word approximations and who have not yet developed full verbal spoken speech. This includes some individuals with autism spectrum disorder, Down syndrome, as well as other neurodevelopmental differences. These individuals communicate in many different ways, including through nonverbal vocalizations, gestures, and communication devices like tablets. Vocalizations in particular are understudied and are not incorporated with currently available communication technologies. Vocalizations are rich in communicative and emotional information, and they are often understood clearly by close family members, but they can be more difficult to understand by someone who doesn't know the communicator well. To give you a sense of what nonverbal vocalizations sound like, I'm going to play samples from 2 communicators that convey delight, frustration, and a request. These vocalizations are important communication, but it may have been hard for you, as someone who doesn't know these communicators well, to understand what each vocalization meant. There is a need and an opportunity here to improve our understanding of these nonverbal vocalizations for the community at large. People who know a communicator well often have this understanding already, and we can build from this to help others, like extended family members, new teachers and babysitters, peers, and other community members who may not yet know what to listen for. Our goal is to develop technology to help the community at large better understand nonverbal vocalizations. For this work, capturing organic, motivation-driven communication in real-world settings along with personalized labels is critical. The personalized labels are important because each communicator may use vocalizations differently. For our first prototypes, we developed a novel, scalable data collection platform with a small wearable recorder and a custom app for labeling vocalizations in the moment.
We use the labels from the app to help isolate the vocalizations from the messy real-world audio that we get from the recorder. This process creates vocalization segments that are labeled by function for each participant. What you see here are 2 different example vocalizations for one participant, one for a type of expression called self-talk, and the other for an emotional state called dysregulation. As you just saw and heard with the audio clips at the beginning, these vocalizations differ by vocalization type and are different for each person. In order to differentiate between the vocalization types, we create personalized machine learning models for each participant. You can see here an example of a 5-way classification model, differentiating between 5 different vocalization types for one participant. Our models produce multi-state classification results consistently above chance. The performance does vary from person to person, and we're developing an understanding of how and when these models work so that we can employ the optimal techniques for machine learning and deep learning for this unique population. We are currently in early pilot testing with the next generation prototype that integrates the full pipeline of Kamala into one app. First, the users can record and label vocalizations in real time using just the app, so there's no more wearable recorder. Then, the users can playback or review the labeled vocalizations immediately from the menu bar. These vocalizations are then sent to us on the back end so that we can build personalized classification models for that user. Once we have enough vocalizations for a reliable model, the classification feature of the app can be unlocked. By pressing and holding the microphone button, the phone captures a vocalization in real time. The vocalization can then be classified using the personalized model we created, and a result can be presented to the user, ideally helping a new communication partner. Again, someone who doesn't know the individual very well, like a grandparent, new teacher, or community member, to acknowledge the vocalization as part of a dialogue and respond accordingly. Finally, the Kamala app incorporates a vocalization library that includes all the different vocalization segments for that communicator. They're organized by type, such as sounds labeled as frustrated or delighted or dysregulated. For people who are not familiar with nonverbal vocalizations as a robust form of communication, simply listening to these sounds in the aggregate can help them learn to hear and respond to these vocalizations as communication. We're in the early prototype and evaluation stage of this new app and are actively working to understand the feasibility, accuracy, and usability of this technology. Based on our early results, we are optimistic that this work can lead to new, impactful, vocalization-based communication technology that someday could truly enhance communication for all. Kamala has been a wonderful joint effort between myself and Jaya, along with our many amazing collaborators and funders. We welcome your emails with any questions or comments or ideas, and we really look forward to chatting with you more during the breakout session. Thanks so much.
Thank you very much, Christy and Jaya, for that great talk. While they were talking, I got a number of emails from people who said, well, we don't eat chicken, we're vegans. So I just wanted to tell you, you can absolutely use that recipe, just substitute for the chicken. You could use cooked beans, cooked lentils, any strongish vegetable, cauliflower, broccoli, squashes, works just as well. And, if you're really rushed for time, like you're a grad student, you don't even have to chop up the onions and the garlic. Just use onion powder and garlic powder. It'll work just fine. Okay, so now on to talking of mixtures. Our next speaker, who also has come up with a very interesting concoction, and that is Hyun-Woo Yuk. Hyun-Woo is a postdoc in mechanical engineering, and he's going to tell us about his bio-based adhesive for wound and tissue sealing. Please welcome Hyun-Woo.
Thank you for introduction. My name is Hyun-Woo from Team Santa Hill, and today I would like to introduce our technology and our vision to novel nature-inspired biodiesel for tissue sealing. First, we want to go for the problem that really excited us, which is bleeding. Human body is full of blood vessels with the flowing blood, so the bleeding is an inevitable challenge in injury and surgical procedures where the cutted part of our tissues bleed because it really disrupts the blood vessel, which is very dense. So this is a ubiquitous and integral part of surgery. It's a very exciting problem for us to take a look deeper. So when we take a look at the surgical landscape and market around this problem of bleeding, there are a lot of surgeries in US, over 5 million per year in major surgical categories. And surprisingly, average 30% of these many surgeries experience intraoperative bleeding-related complications. So this is a very common problem. And also it's not surprising that some form of the hemostatic agent to control these bleedings are already having a high, very high adoption rate, as over 50% in solid organs such as liver, and even over 80% for spine surgeries. So this hemostatic agent to control bleeding is already forming a total addressable market of $1.5 billion per year, which is a very big market, which is good news for commercialization. So this is a ubiquitous problem. So it's not surprising there are conventional techniques to address this bleeding problem, such as compression with gauzes and just suturing up. which oftentimes not ideal. So there are other hemostatic agents that from biomaterial to coagulation factors to adhesives from various companies. You might identify some familiar names. But what is interesting to us is a hemostatic performance, especially for the criteria that might be important to save people's life in a very serious situation. So the questions are whether rapid hemostasis within 30 seconds, that is fast enough the surgeon can feel rapid, will be addressable, whether severe bleeding that losing a lot of blood every second will be addressable, or anticoagulated or coagulopathic patient bleeding will be addressable, which is common in various diseases and surgical operations. So surprisingly, the answer to this criteria is all no for existing product and techniques. And as a result, what is happening is, despite the use of hemostats, still 32 to 68% of intraoperative bleedings are uncontrolled. So which is huge ongoing problems. It's costing tons of lives out there. So it is really important for us to have something to convert this uncontrolled situation to control. So this video is really capturing the situation a little better. This is a heparinized porcine blood, which cannot form a blood clot spontaneously. This is severe bleeding, losing a lot of blood every second. But as you saw in the previous table, there is no existing product or solution that can form rapid hemostasis for this bad situation of anticoagulate and/or severe bleeding. So that's exactly what we want to solve with our technology, Biodiesel Paste, which is a preparation-free and simple-to-use paste that can be applied on gauze for easier handling for surgeon or directly on a bleeding surface. Just simply press for 5 to 15 seconds with a gentle pressure, no preparation, no mixing, nothing else, but just press for a few seconds, and that's it. It immediately forms the severe bleeding stop. And it requires no blood coagulation, so the coagulopathic or anticoagulant patient would see the same efficacy and is very robust. It really resists the burst from this organ-deforming high pressure we artificially introduced. So it is so exciting for us. So we tried this on pig with our Mayo Clinic collaborator recently. This is a fully anticoagulated pig with a very sizable big deep injury and blood is gushing out, you can see. We first tried a very popular FDA-approved Taco-Seal, as fibrin patch, expensive one, and compressed that on this injury for 3 continuous minutes following the manufacturer's guideline. And after 3 minutes of continuous pressure, it's on the liver. It is actually, you can see that bleeding still comes happening below the patches and eventually accumulate and leak out sideways. So it literally didn't stop the bleeding. So this is a hemostasis failure cases. So which is very bad news for our poor piggy. So we wanted to save her. So we removed this failed hemostatic patch from the surface of the liver and see this continuously bleeding liver surface and tried our paste on top of it to try to rescue it and just applied a 15-second gentle pressure to be conservative and did nothing else. No preparation, no mixing, nothing else. And just a check after 15 seconds in a single trial addressed this severe anticoagulate bleeding, significantly outperforming the Taco-Seal, which is exciting to us. So what we are now looking for in terms of timeline and milestone is we are looking for a seed investment and spin-out from MIT, hopefully 3rd quarter of this year, to do a tech transfer and product development in startup toward the ID preparation, maybe with a little more funding for the first in-human pilot trial to 1.5 to 2 years from now on. And to do that, what we are looking for now is investment for seed and Series A investors to continue our exciting journeys and talents to team up together to form a strong team to work on these challenging problems and collaborations from clinical and corporate peoples to maximize the potential of this interesting technology to save people's lives in the future. With that, I would like to thank you for your attention and please ask questions in the breakout sessions. Thank you.
Well, thank you very much, Hyun-Woo. And our next speaker is part of the Technology to Improve Ability program. And I'd like to introduce Ravi Rasalingam. Ravi is an instructor at the MIT Institute of Medical Engineering and Science. And he's going to tell us about the novel device they've been developing for obstructive sleep apnea for people with Down syndrome. Please welcome Ravi.
Thanks very much, Leon. Hi, My name is Ravi Rasalingam, and I'm a physician at VA Boston Healthcare and an MIT Catalyst Fellow. On behalf of the Roche Lab, I'm excited to present to you our project, a novel device for treatment of obstructive sleep apnea in people with Down syndrome. I would like you to imagine holding your breath for as long as you can, fighting that growing urge to breathe until suddenly gasping for air, and then imagine repeating this over and over again, multiple times per hour. Obstructive sleep apnea, or OSA, describes a condition where this happens due to the episodic collapse of the muscles of the throat. The only difference is that you're asleep, blissfully unaware of the damage this is doing to your body. These obstructions to breathing, or apnea episodes, typically occur at the back of the throat and involves the tongue and the soft palate, as marked by the X in this picture. Frequent apnea episodes result in disrupted sleep and daytime sleepiness. OSA is extremely common in a genetic condition called Down syndrome. OSA affects 30 to 50% of Down syndrome children and 90% of adults. This is thought to be related to a reduction in the size of the bones of the face, causing a tongue that already has less muscle tone to occupy more space in the mouth. Disruption of sleep causes significant cognitive decline, in particular, the development of executive functions such as planning, problem solving, and emotional control. This ultimately affects the ability to progress to independence. It is also associated with increased risk of cardiovascular disease, and in particular, a rise in the pressure in the lungs called pulmonary hypertension. OSA is also common in the general population. 17% of men and 9% of women aged 50 to 70 have moderate to severe OSA in the US. It is very likely that you already know someone who suffers from this condition. In addition to daytime sleepiness, OSA has been associated with an increased risk of heart attack, stroke, and premature death. There are treatments that are available that can prevent apnea episodes. The most commonly prescribed is called continuous positive airway pressure, or Cpap. This describes a device that forces air continuously through a wide-bore tube and a tight-fitting mask that fits over the nose or nose and mouth. Much less commonly used is the mandibular advancement oral device, which is a mechanical hinge that forces the lower jaw forward and therefore the tongue, relieving the obstruction at the back of the throat. As you can imagine, these solutions are uncomfortable. Cpap is the current gold standard treatment. However, only 17% of Down syndrome OSA patients prescribed Cpap have a mask that fits and use the device for greater than 4 hours per night, while in the general population, only 60% of OSA patients tolerate Cpap. Our approach, therefore, has been to develop a device that not only prevents apnea episodes, but is comfortable enough for a patient to use on a regular basis for a prolonged period of time, something that is necessary to treat a chronic condition. This focus on comfort and usability is even more important when considering a device for children. Our device uses a custom-fit dental aligner that not only goes over the teeth, but extends back across the palate and allows for the transmission of intraoral suction to stabilize tongue position. Low-level suction is used to stabilize the tongue and is similar to what you may have experienced when visiting a dentist. Ultimately, this prevents obstruction and promotes nasal breathing, which is the preferred route of respiration. Dental aligners are widely known and a highly successful established method for orthodontic treatment. The technology is advanced to such an extent regarding comfort and discreetness that people forget that they're even wearing them. We have leveraged these advances and have used this technology as a platform to add a method to stabilize the muscles of the mouth and the throat. Digital intraoral scanning is a technology that has revolutionized orthodontic treatment by allowing a quick and accurate impression of the oral cavity to be taken in minutes. This is especially important when treating children. We have used this technology to understand the individual nature of how the palate interacts with the tongue and have developed insights that form the basis of a unique design and approach for treating OSA. Our device solves the issues that have prevented Cpap from meeting the needs of a significant proportion of OSA patients. As we use suction rather than forced air, our device does not need a tight-fitting mask in order to be effective. Discomfort from the mask is the most common cited reason for the inability to tolerate Cpap. The level of suction used by a device is low and can be transmitted through a small, discrete tube rather than the monster tubing that Cpap uses. This allows us the opportunity to miniaturize the size of the pump and its components into a wearable form factor. The overall impact of our device on the sleep environment is minimal. Our approach is poised to have a significant impact on a large number of people. The OSA therapeutic device market is large and well established. The disease is common, with almost 6 million people diagnosed with OSA in the US. The current total market value is in excess of $3 billion. This market is also expected to grow based on changing demographics and a rise in risk factors such as obesity. There's also a general increased awareness and capability to detect sleep disorders in the community. Our ability to gain traction in this large market will allow the opportunity to focus on specific populations where the prevalence of OSA is especially high, such as Down syndrome. We have filed a provisional patent for our technology and have identified that there's already a regulatory precedent that guides the evaluation of intraoral sleep devices. In addition, reimbursement codes for intraoral sleep devices already exist for private insured and Medicare patients. As participants in the MIT iCore Customer Discovery Program, we have learned that there's already an established pipeline for delivery of OSA treatment devices. Based upon our interviews with sleep specialists, the main problem, however, has been a lack of effective treatment tools that are comfortable enough for nightly use. We envision our customers will be sleep physicians who traditionally have been the decision makers regarding treatment choice. In addition to the standard paradigm, we believe our approach, based on a dental appliance, enables a second market pathway that involves partnering with dentists specialized in sleep disorders. We have made a tremendous amount of progress over the last few years, from identifying and validating a need, conceiving a solution, and fabricating a prototype that we have tested in healthy volunteers for proof of principle. We are currently optimizing this prototype for a planned clinical study in OSA patients later this year. Our project has benefited from the generous support of a number of funding organizations. We have also built our team to strategically take advantage of skill sets necessary to accelerate our progress. Our goal is to establish a company once we've demonstrated the clinical efficacy of this approach. We are seeking interested parties, especially those with expertise in regulation of medical devices, as well as management expertise. We also welcome funding organizations willing to support our project. We have a tremendous team that includes MIT postdoctoral and undergraduate engineering students, sleep physicians, researchers, and dentists, as well as programmatic clinical and technical advisors from the wider MIT and Boston healthcare innovation communities. We hope you will join us in our breakout room and in our goal to get rid of the mask. Thank you very much.
Thank you, Ravi. Our next speaker is Rebecca Kleinberger. Rebecca is a postdoc in brain and cognitive sciences at MIT, and she's going to tell us about her real-time speech modification system to enhance fluency in people who stutter. So please give a big welcome to Rebecca.
Thank you, Leon. Mumble melody. Stuttering is often portrayed negatively in popular culture, from Harry Potter, Marvel, Star Wars, Winnie the Pooh. And it's pretty unfortunate for people who have to deal with the stigmatization, because here's what people who stutter actually look like. You may recognize a few famous people who stutter, but mostly this is people like you and me. It's an invisible struggle. And it touches 1% of all adults and 8% of children. And there is no cure. And here's what stuttering sounds like and feels like.
I had speech therapy since Harsh, the person I'm working with, is like she specializes in she specializes in stuttering.
And as you can imagine, stuttering often has huge impact on social interaction, quality of life, and economic productivity. And it impacts so many parts of life because it impacts communication and sense of self-worth. There are a number of approaches and treatments that have been tried to help, and an ideal treatment would need to be effective, accessible, and low cognitive load. And one of the most effective is still speech therapy because it's very personalized, but it can be quite expensive and not always accessible. And our system really doesn't try to compete, but really to work alongside with speech therapy. There are also some hearing aid devices that exist on the market that take the voice and add a delay or a pitch shift to the voice. And they can be quite effective, but they're quite expensive. And finally, there are some apps that exist that do the same transformation, but that are often not very effective because they're not personalized. And after a few weeks or months, the brain gets used to it and we start stuttering. Our solution is Mumble Melody, and here's how it works. When someone speaks, we take the sound of their voice and we transform it in real time into a musicalized version of the voice that we then feed back to the speaker. And when we did that, here's what happens. And this is the same person as earlier.
The Notes app I use all the time because if I think of something, I'm going to forget it, so just write it down. Or like little things that I see that I want to look into later, I write it down.
So how does that work? Well, to simplify, when you speak, there are really two signals happening in your brain. There is what you plan to say, some kind of inner voice, and then there's what you actually say. That's your outer voice. And for people who stutter, there seems to be an issue with how those two signals get compared in the brain. And by providing this musical feedback, we avoid the speech neural pathway and we leverage the music processing part of the brain. Also, music is so rich and contains so many parameters that our system can be very personalized to the individuals. So we use machine learning to optimize to the user and the specific time of use. And thanks to that, the brain doesn't get used to the signal. And our prototype is already available on the App Store, so people can download it and use it. And we're already collecting data to help us refine the product. We are very focused in how the technology will be used in the market and who are the gatekeepers. So we're in touch with a lot of people in the value chain, starting with local patient support group, the NSA, National Stuttering Association, as well as ASHA, the American Language Hearing Association, and some local therapy center like the Siskin Stuttering Center, as well as neurologists from UCSF and MGH, and the Society for the Neurobiology of Speech. And here is our team. So we are experts in neurology, clinical medicine, software, voice, music, and experience design. And here's what we're looking for. We're looking for team members, especially people with business expertise. We're looking for people who stutter. If you stutter, or if you know people who stutter and who would be interested in collaborating and participating in our study. We're looking for connection with foundation and industry partners. And finally, we're looking for sponsors to make this whole project happen. Thank you very much.
Well, thank you very much, Rebecca. Great talk. Our next speaker is Dana Al-Sulaiman, and Dana is a postdoc in chemical engineering at MIT, and she's going to tell us about multiplexed measurement of miRNA in tissue sections. Please welcome Dana.
Thanks for that introduction, Dion. Today, there are many cytotoxic agents and targeted therapies available for cancer, but still many patients develop resistance to therapies. This ineffective treatment can not only lead to a significant economic burden, but also to the development of much more aggressive cancers. One of the main reasons for this therapeutic resistance and poor patient survival is a phenomenon called tumor heterogeneity, one of the main challenges facing clinical oncology today. So say I have a bunch of apples and one of them is rotten. I take a snapshot and ask you to describe the photo. A traditional bulk bioassay will give me this information, essentially an average of all pixel values. A single cell bioassay might give me much higher resolution, but without any contextual information. What we are developing is a spatially resolved bioassay that can retain this contextual information. So why is this important? Just like this bunch of apples, a tumor is heterogeneous, And this variability makes it difficult for treatments to eliminate the entire tumor. So what is the spatially resolved information we are detecting? It comes from a highly promising biomarker for cancer called microRNA. These are short non-coding RNA that have very important gene regulatory functions. But in cancer, their levels get deregulated through either overexpression or underexpression of the microRNA in the tumor compared to normal tissue. Now, it turns out that just detecting the levels of microRNA is not enough to make informed clinical decisions. There are two further requirements, and the first is spatial information. This study has shown that detecting low levels of microRNA, specifically in the stroma of the tissue, predicted a positive patient outcome. However, if it was detected in the tumor, it would predict a poor patient outcome. Now, other studies have shown the same thing, that the location of microRNA in a tissue is very crucial to making accurate clinical predictions. Now, the second requirement is multiplexing. That is the ability to detect not one, but a whole panel of microRNA. And this allows you to differentiate between normal and cancer tissue, but importantly, to distinguish between benign and malignant tumors. Now, unfortunately, there are no current techniques that can simultaneously satisfy both criteria. A bulk sample bioassay may offer multiplexing, but it lacks spatial resolution, and a spatially resolved bioassay such as FISH offers limited multiplexing and long, laborious workflows. There is thus an unmet clinical and technological gap to be filled, and for this, we propose our project. We have assembled a team of microRNA experts from BIDMC, headed by Professor Frank Sack, as well as a team of experts in microtechnologies and biosensing from MIT, headed by Professor Patrick Doyle. And together, we developed a technology for spatially resolved and multiplex microRNA detection, all in a format that fits perfectly into the standard pathology workflow. So you start with a formalin-fixed, paraffin-embedded tissue, and this is the standard, with no sample preparation, short assay times, and a single fluorophore is required. So how does this work? It's based on a nanoliter well array, and you can think of this as a densely packed grid of square wells. Each well is like a pixel in an image, and the wells are filled with microRNA detecting hydrogel posts, shown in blue, as well as lysis reagents. The posts are functionalized with DNA probes targeted towards your microRNA of interest. Now, what you can do is press this array against the tissue section. The lysis reagents will digest the tissue, releasing the microRNA, which is then captured by the posts in a spatially resolved manner. So how would this work in practice? What I've got here is an H&E stained tissue section from a genetically engineered mouse model for non-small cell lung cancer. And a pathologist would be able to identify the 3 tumor regions as labeled herein, 1, 2, 3, as well as the normal adjacent tissue. But all 3 regions look exactly the same, and there are no further information apparent in order to inform clinical decisions. Our platform is able to provide this information. And here's just a fraction of it. For example, seeing that microRNA 21 is overexpressed in region 1, a clinician might prescribe treatment A. But now seeing that microRNA 19b is overexpressed in region 3 might indicate resistance to this initial treatment. So then a clinician would prescribe treatment B instead. And now combining all this information, a clinician would be able to devise the most appropriate and personalized treatment strategy effective against eliminating the entire heterogeneous tumor. Overall, our platform represents the only spatially resolved and multiplex microRNA bioassay. We believe it can be used by researchers to better understand tumor heterogeneity, but also by clinicians in order to devise the most informed treatment plans. We're now working with our clinical collaborators at the IDMC on a human breast cancer study, and in the future, we'd be interested to talk to potential investors as well as companies with synergistic technologies. With your support, we hope to tackle one of the major challenges facing clinical oncology today, leading to much better patient outcomes and a lower economic burden. Thank you all for listening.
Well, thank you very much, Dana. And our next speaker in the session is Ray Liao. Ray is a PhD in electrical engineering and computer science. And he is going to talk about the non-invasive assessment of pulmonary edema using machine learning. Please give Ray a warm welcome.
Greetings, everyone. My name is Ray. I'm a PhD student at MIT CSAIL. I work with Professor Paulina Golan at MIT, and Dr. Stephen Horn and Dr. Seth Berkowitz from Beth Israel Deaconess Medical Center. We developed novel machine learning algorithms to non-invasively assess pulmonary edema in order to help physicians better manage heart failure patients. Dr. Stephen Horn is an emergency room physician. 2 years ago, Dr. Horn reached out to our lab at MIT and told us how difficult and frustrating it is to manage heart failure. 20% of heart failure patients get readmitted within 30 days of discharge. Dr. Horn is not alone. As the leading cause of hospitalization, every year there are 1 million heart failure hospital stays in the US, and the healthcare system spends $12 billion on heart failure hospitalization. Most of the heart failure emergency room visits and hospitalizations are due to patients' shortness of breath, caused by their fluid overload in the lungs, aka pulmonary edema. When the heart cannot pump out blood effectively, the blood pressure backs up in the right ventricle all the way to the lungs. Due to the increased pressure, the blood and the fluid leak into the lung space, causing pulmonary edema. In clinical practice, chest X-ray is commonly performed to assess pulmonary edema and is a routine examination in the emergency room to make differential diagnosis. However, the reading and interpretation of chest X-ray images suffer from high inter-reader disagreement among radiologists. This inconsistency leads to poor discharge and disposition decision-making. Many heart failure patients are discharged from hospital while they still have fluid overload. Those patients eventually exacerbate again and get readmitted to the emergency room. This clinical challenge, on the other hand, creates an opportunity for machine learning and AI to pick up those subtle yet important pathological findings from chest X-ray images that might have been missed by clinicians. To develop such a robust machine learning model, we have worked with Beth Israel Deaconess Medical Center and have collected about 370,000 chest X-ray images and their associated radiology reports, written by many radiologists. The radiology reports have documented the interpretation of those images. We have constructed an AI model that is trained from the images as well as the reports to learn the assessment of pulmonary edema. This approach enables our computer vision model to essentially acquire the capabilities of many human experts. When a new chest X-ray image comes in, our AI model takes advantage of the aggregated capability of chest X-ray interpretation and instantly makes accurate assessment of patient fluid status. Our experimental results that are recently published in a top clinical journal demonstrate that our AI model is comparable to the performance of 4 radiologists' consensus assessment and outperforms the independent assessment of each individual radiologist. Our recent retrospective analysis also shows that the fluid overload that our computer vision model is able to detect correlates with patients' likelihood of getting readmitted based on the clinical data collected at Beth Israel over 10 years. We are very excited about the potential of our technology and also the future of computational intelligence in medicine. The instant and accurate radiographic image interpretation that our AI model offers can improve hospital efficiency. By picking up missing pathological findings, our algorithms help physicians stage early interventions and make better discharge decisions. to reduce avoidable readmissions and emergency room visits. With the healthcare system shifting to value-based care, this technology lowers the cost for both providers and medical insurers, and eventually improves clinical outcomes of heart failure patients. Sparked at MIT, incubated in the DISH-funded program, we are looking to make an impact in medicine. We are currently working with 3 clinical centers to generalize our AI model and pilot our AI system, including Beth Israel Deaconess Medical Center, Tufts Center, and University of Colorado Hospital. Also, Mayo Clinic, Piedmont Healthcare, and Veterans Affairs Hospitals in Texas have agreed to partner with us. Our toolkits to build this initial AI solution and our channels to hospital information systems can be modularized and readily scaled to other clinical domains. With the rapid digitalization of healthcare data over the past decade, hospitals have accumulated a large amount of clinical data that has yet to be utilized. Retrospective medical data contains invaluable insights for future clinical management. We will ultimately make it faster and easier to discover those patterns and to build computing solutions upon them. We are at the early stage of this journey. If you are excited about this future as we are, please come to our booth and share your thoughts. Thank you.
Well, thank you very much, Ray. And I'd like to thank all of the speakers in this session for doing such a fantastic job. And it's now time for our second breakout of the day. And this is where you get to interact with these really bright, enthusiastic people and share your comments with them and find out more about what they're doing. As we did in the first session, we're using the Hopin Expo booths for those of you that might have come in late. What you do is you look at your Hopin screen, you'll see on the left it says Expo, just click on that and it's going to bring up a whole list of different booths. When I did this earlier, it did show everything from today and tomorrow, so you'll have to sort of go through and find the particular projects you want to talk to. Click on those, you'll then enter the booth. If you want to, you can turn on your audio and video to have a conversation by hitting share audio and video. You can also chat to them using the chat function in the Hopin Expo booth. There can only be 10 people on audio and video at the same time, although lots of people can be in the booth. So if all of the on-camera slots are taken, just wait a few minutes for someone to wrap up their conversation and then request to appear on camera again by clicking the share audio and video. And if you can certainly go from one booth to another if there are multiple booths that you want to visit. So we're now at about 1:18. And what we'll do is let's meet back here at about, let's say give it just about 22 minutes. That will take us to about 1:40. So we'll meet back here about 1:40. And at that point, just click on stage on the left on your Hopin screen and come back and we'll wrap up at the end of the day. So looking forward to seeing you at about 1:40. Thank you all. Well, hi, everybody, and welcome back. We decided to let the breakouts run for another 5 minutes, which is why we're sort of 5 minutes later than I'd said, because there seemed to be a lot of good conversations going on. So I hope you enjoyed all the sessions today. We started off with, great panel with Christine and Rich, and we'd like to thank them very much. Also, thanks to all of our speakers for doing a great job. Thanks to all of you for attending today. And we will have the a recording of today's idea stream that'll be online in about a week or so. And a quick recap: if you can help any of our teams, please do so in whatever way you can. Either contact them directly or just reach out to the Despande Center, email us, and we can make the connections. Any corporations that are interested in getting more engaged with the Despande Center, finding out about our corporate program, please just contact us and we will get back to you. For people who like what they saw and would like to support us financially, there's a link at the top of our webpage where you can give to the center. And certainly, we're looking forward to doing this again in person next year. I am looking forward to seeing everybody in the fall when hopefully we're all back on campus. And if you have not yet been vaccinated, I hope you're able to get a vaccine soon. Let's say my advice is don't plan any activities for about 2 days after your second shot. Certainly a wise thing to do. And if you're in the Boston area, enjoy the rest of the afternoon. If you're somewhere else in the world, maybe it's evening, enjoy the evening. Tomorrow is going to be our Physical Sciences Day, and we have a fantastic program, also another very interesting panel. So we look forward to seeing you tomorrow, and take care and look after yourself.